Hello everyone, my name is Allison Ludwig. Am I on? Can you guys hear me okay? Okay. So I'm Allison Ludwig. I'm a third-year PhD student in the Department of Comparative Biomedical Sciences, and I love photoreceptors. Now, photoreceptors are the cells that live at the back of your eye in a tissue called the retina, and you should also love them because they're able to capture light, and all of you are using them right now to see me standing in front of you. Now, there are millions of people in the world who are losing their vision right now because their photoreceptors don't function properly. And because there's nothing we can do to stop that from happening, their photoreceptors actually die. And once your photoreceptors die, you can't see anymore, and you go blind. Now, my lab was one of the first in the entire world to show that we could take a stem cell, which can become any cell in your body, and in a dish, we could turn that into a photoreceptor. And so our question naturally was whether we could take these photoreceptors that we can grow in a dish and use those to replace the ones that people are losing due to retinal disease. Now, quick show of hands, how many of you would be willing to let me experiment on your eyes to see if these cells work? Anybody? No? That is good. So the Food and Drug Administration requires that we make sure that these cells are safe and effective before we take them into human patients. And so we turn to animals who have the same type of retinal diseases to answer questions that we have about how these cells work. Now, specifically, I'm interested in taking these cells and figuring out how we get them into the eye and what they do once we can get them there. So I started off by drawing these cells up into a little syringe and just injecting them into the eye. And guess what we found? Pretty much all of the cells died, and none of them stayed where we put them. So, as science goes, we went back to the drawing board with a couple of engineers here at the WID, and we tried to find a better way to package these cells to protect them and keep them where we put them during surgery. And so what we were able to design was a tiny scaffold that can fit on the tip of your finger and is thinner than sheet of paper. And it can actually protect those photoreceptors during transplantation, and that's what we found. That when we package these cells into this tiny scaffold, they would stay where we put them and they would be protected during surgery. So for the rest of my thesis, we're trying to use these tiny scaffolds to see what happens when we put those cells in the retina and what effects they have on vision when they get plugged into the rest of the cells that are there. And what we're hoping in the future is that we'll be able to scale up these scaffolds to use them in human clinical trials someday, with the ultimate goal of restoring vision to those patients who've lost vision and don't currently have photoreceptors. Thank you.